Richardson, and I am a researcher and teacher for the Center for Sustainable Development. I am from Ottawa in Canada and have a Bachelor of Science in Biology, and my research focuses on the health and toxicity of agriculture and aquatic ecosystems.
Hi everyone, I'm Mark Young and I am the research teacher at CSD for Permaculture and Sustainability. I have a degree in botany and zoology with several years of experience in agriculture, horticulture, and a passion for microbiology. And without further ado, we'll hand it over to the Biochar Biothird team.
I'm Gabby Quintero.
Jada Rudolph.
Jordy Bertrand. Max Nonamacher.
Claire Cornelius. Maddie Ruby.
And I'm Max Briska.
And we are evaluating the germination and growth of cowpea amended with biochar, inoculated with fermented organic seaweed liquid extract. Many small island developing states, also known as SIDS, have sandy soils. Sandy soils have a low cation exchange capacity and a low carbon content. What this means is that the sandy soils aren't able to absorb and retain things such as nutrients, water, and microbes. Because of these two properties, farmers tend to over-fertilize their soils. Over-fertilization and eutrophication have many negative impacts on marine ecosystems downstream. In addition, sandy soils contribute to food insecurity because many farmers are unable to grow their preferred crops.
Okay, so biochar. Biochar is a stable carbon matrix. So to break that down, it has several carbon layers which form together to create a matrix. And that matrix is stationary, so it doesn't break apart or decompose or deteriorate. Biochar also has a high cation exchange capacity. This means that it is able to absorb lots of nutrients, water, and microscopic organisms. Biochar also has a high carbon content. So this is good in relations to dealing with sandy soils, as Gabby mentioned, because it has such a low cation exchange capacity. So coconut husk biochar, also known as CBC. We use coconuts to make our biochar because we have such an abundance here in the Bahamas. It takes around 40 to 50 years for a coconut just to decompose. So it's really good if we use it to make our biochar process. So in order to make biochar, we start off by drying our coconut husks out in the sun. After that, we use it through a process called pyrolysis, which is thermally decomposition in a low oxygen, high heat environment. So we put the coconut husk in the kiln, as seen in the second picture, at 500 to 400 degrees Celsius for 4 to 6 hours. After that process is done, it then comes out looking like the last picture, and that is our coconut husk biochar. With that, we can grind it up like Abby is holding and mix it with our soil for the best results.
So, biofertilizer. Biofertilizer is an effective, non-polluting, non-toxic form of fertilizer. It's used as an alternative to synthetic fertilizers, which can cause several environmental and health hazards. So biofertilizer contains macro and micronutrients, growth regulating hormones, and because biofertilizer is literally living, it has microbes. Biofertilizer has been proven to not only improve germination rate, but the resistance and yield of cover crops, as well as production crops. Biofertilizer is also used to improve soil texture and moisture retention, as biofertilizer can decompose biomass and therefore can be used in conjunction with compost. This is a picture of the biofertilizer that we then inoculated into our biochar. So for our biofertilizer, we decided to use Sargassum fluitans, which is more commonly known as seaweed. Like coconut husk, it is very, very abundant in the Bahamas, so it was very good for us to use in our experiment, as we wouldn't be taking many resources from our ecosystem. So we put our sargassum, which as you see here in the top right picture, through a process called anaerobic fermentation. Basically what that means is we take the sargassum off the beaches and place them in airtight containers that have no oxygen and leave it for 4 weeks. Through that period of 4 weeks, the seaweed begins to decompose and microbes and bacteria start eating away at the seaweed, which can then be used in our soil. So this is a picture of our biofertilizer, what we call seaweed liquid extract, or SLE, that is very, very concentrated, and we measure it and then dilute it with water. So biofertilizer and biochar have been proven to work as amendments for soil individually, but for our research project, we have decided to use them in conjunction with one another and test the results.
So the objective of our research is to figure out what concentration of SLE inoculated with CBC will provide the best germination, growth, and yield of cowpea while it's growing in sandy soil environment that lacks nutrients that these plants need to grow. Our experimental design, we used a randomized block design to account for any environmental variables that could skew our data. We have 9 treatments ranging from 20% SLE to CBC to 0.5% CBC. For our 7th treatment, we used Miracle-Gro, which is a synthetic fertilizer at a concentration of 0.4%, which is the recommended amount Miracle-Gro says we should use. For our 8th treatment, we used plain CBC inoculated in the soil as a control, and our 9th treatment, we had just plain sandy soil. We have 3 replicates for each treatment to ensure that if anything should happen to one of the treatments, one of the replicates would ensure that our data would not be skewed. We have 128 seedlings per tray, meaning that we have 3,456 seedlings in total for our study. In this top picture here, we are mixing our CBC and SLE into the field at Pineapple Fields Resort as our field experiment. And this bottom picture is us a week later at the same field weeding.
Our indicator species is cowpea, or Vigna unguiculata. And we chose cowpea because it's a hardy cover crop, which means that farmers grow it in between production crops to add nutrients, nitrogen, and nitrogen-fixing bacteria into the soil. Cowpea was practical for our experiment in particular because it grows well in harsh environments, sandy soils, and nutrient-poor soils, which is what we have here in the Bahamas. Cowpea is also practical economically because it feeds both animals and humans. On the first 7 days of our study, we tested for germination rate, and on the last day, we tested for germination success by counting the seedlings. And for our shoot and root links, we used a program called ImageJ. So what we did is we took our plants out of the soil in the trays, and we put them on a white sheet of paper, as you can see here, with a ruler on the side. And what we did was we drew a line from 1 centimeter to the next, and then we drew a segmented line from the top of the shoot to the base, and then from the base to the root to measure our shoot and root lengths. The reason why we used this program in particular is because we couldn't measure all of our plants because we had such a big sample size. And so the sample size we measured for these pictures is 810 plants. We used a statistical program called R3.2.4, which is a generalized linear mixed effects model. And we used this program in order to find out if our data was statistically significantly different. And we couldn't use a simple t-test or ANOVA because the scope of our study was so broad.
At the beginning of our experiment, we hypothesized that an increasing seaweed liquid extract concentration would produce increasing germination and growth of cowpea. Our predictions were high concentrations would produce low germination and growth, low concentrations would produce increasing germination and growth, and our optimal concentration would be between 2.5 and 5%. Table 1 demonstrates the physiochemical properties of seaweed liquid extract. Our pH, as you can see, stayed relatively the same throughout all of our treatments. We started with our stock treatment of 100%, and our electrical conductivity of our 100% stock treatment was relatively high compared to our other treatments. This is due to high nutrient and salt levels. As we diluted each treatment, you can see here that the electrical conductivity went down dramatically until we got to our Miracle Grow. Table 2 represents the physiochemical properties of our soil with biochar and seaweed liquid extract through 21 days. On day 0, our pH stayed relatively the same, as well as our electrical conductivity. On day 21, you can see that our electrical conductivity went down dramatically. This is due to either plants absorbing all the nutrients or rainfall runoff. Figure 1 demonstrates all of our treatments, germination rate, and success. Our x-axis is our days after sowing, and our y-axis is our germination rate and success. The next few slides will be the same figure broken down to help you better understand. Figure 1A demonstrates our control compared to our high concentrations, which are between 5 and 20%. As you can see, our red line is our control, and our high concentrations did not do better than our control treatment. Figure 1B is our control compared to our low concentrations, which are between 0.5% and 2.5%. All of our low concentrations did not do better than our control except for our 1.25%. Figure 1C demonstrates our control compared to our sandy soil and our Miracle Grow treatments. Both sandy soil and Miracle Grow did better than our control treatment.
Here we have a box plot of our results for our shoot lengths of all 9 treatments compared to our control. We can see, marked by the asterisks, those are points of significance in our data, and marked by open circles, those are outliers in our data. We can see that 0.5%, 5%, 20%, and Miracle Grow, concentration of 0.4%, all had significantly shorter shoot lengths when compared to the control. Sandy soil, however, had significantly higher shoot lengths when compared to the control. Here's another box plot of our 9 treatments compared to our control in terms of root length. Throughout all 9 treatments, there are fairly similar root lengths when compared to the control, except for the 20% concentration, which had slightly longer roots when compared to the control. So our conditions of our soil, of our treated soil, were measured to have a pH of 8, which meant high salinity and high electroconductivity, as well as high nutrients. So cowpea, the plant we were growing, often prefers nutrient-poor soil, or pHs from 5.5 to 6.5. So when looking at the effects of our seaweed liquid extract on germination rate, success, and seedling growth, we noticed that high and low concentrations of seaweed liquid extract actually inhibited cowpea growth. So in conclusion, high nutrient contents and salinity in our coconut husk biochar inoculated with seaweed liquid extract inhibited cowpea growth. Optimal concentrations of seaweed liquid extract in coconut husk biochar was 1.25%, as mentioned earlier. Cowpea, as I mentioned earlier, again, prefers nutrient-poor soil and therefore preferred our naturally sandy soils with no additives, which actually stimulated its growth. However, although our coconut husk biochar and seaweed liquid extract did not have the effect we wanted on stimulating cowpea growth, cowpea is the ideal crop for small island developing states. It needs little to no nutrients to do very well, and it also can feed animals, people, and can return nutrients back to these sandy, nutrient-poor soils. We will be sending our samples of coconut husk biochar, seaweed liquid extract, and our treated soil to labs to be analyzed, and we will be looking for the macro and micronutrients in all of them. Plant growth hormones included the auxin, gibberellin, and cytokinin. We will also be looking for those in our samples as well. Continuous research will be looking at the crop yield of our plants, as well as the combination of biochar and biofertilizer on production crops. And production crops often need more nutrients to do better, which include tomatoes. Varying compositions of biochar will also be looked into, which include using coconuts themselves, wood, as well as other commercial products. We'd like to thank Mark and Sarah for their help this semester, as well as David Barlin at Pineapple Fields Condo Resort Hotel for letting us do our field study there, as well as CSD for all their help as well. Thank you very much. Are there any questions? Yes.
So the permaculture team talked about pH balance in your tanks. They talked about the lack of potassium, nitrogen, and phosphate. Is there any cross research done in using biochar affected the soil? Because I didn't see if those things
So your question was about how biochar affected
affect the soil in those where there's lack of those things.
Hmm. How biochar affects the soil in nutrient-poor environments.
Yeah, and just mainly the potassium and the
Hmm.
nitrogen phosphate.
So biochar often, it doesn't really affect the soil in any way because it's more absorbing what's already in the soil. So if you load it with seaweed liquid extract, which was full of nutrients, and then put it into the soil, that's how it can add nutrients to the soil. But putting biochar in the soil itself, it will simply absorb the nutrients that are already there because it acts like a sponge. So adding biochar itself won't have any effects, it'll just take away nutrients. Yeah.
Other plants been studied in the same way on this island, so this being a monoculture of cowpeas versus
So we actually had 3 different plants we started with, but cowpea was our end result. And also, we'll be looking into other crops, like the production crops, like I mentioned earlier. But we started with cowpea because it's a cover crop, and cover crops are incredibly hardy, and we thought would be a great starting point because they don't need a lot of nutrients, and we were just curious to see what would happen. But we'll be looking into other plants that need more nutrients to do better.
Yeah, actually, we grew cowpea, buckwheat, and millet in our study, but because we don't have much time here, we only analyzed the results for cowpea. And also, in further studies, we're going to be working on production crops, which are like tomatoes and sweet potatoes, that the Bahamians can export and make money off of.
So the high salinity in our soil is partly because of the calcium carbonate, and there's already a lot of there's high salinity in the soil to begin with, and calcium carbonate really is where the high salinity comes from.
Also, because we use biofertilizer in our biochar, that increases the salinity just because the nutrients in the seaweed itself have a really high salinity, and as well as microbes and stuff like that that do increase the pH level. And for that reason, that's one of the reasons why cowpea was sort of inhibited by higher concentrations, just because by having so much salinity, it increased the pH level to an extent where cowpea was actually inhibited for the growth. Yeah.
Are there other islands or places that are doing the coconut pulp?
So the question was if there are other islands that have used coconut husk biochar. Other places around the world do use biochar. It's commercialized, but it hasn't really been implemented in small and developing states like here in the Bahamas. So our specific study was to use biochar and study whether it could help and improve the sandy soil environment here. But yeah, it's definitely been commercialized, just not in areas like the Bahamas.
Also, a big part of our study was making it accessible to people who didn't have a lot of money. So we wanted to make sure that we were using resources that were cheap. So for example, the thing that we used to put our biochar in, our kiln basically, was from our boneyard. So it was just reused, so we made it for free. And so we want to make it accessible for everyone instead of building a giant commercial plant that really wouldn't help anybody and they'd still have to pay for the biochar. So we want to make it for free.
And that's also why we used coconuts and sargassum, because they're incredibly abundant on this island. And if a local farmer wanted to repeat this process, it would not be very hard to come by these resources. to make biochar and biofertilizer.